Myself, Dr. P. Samuel, Assistant Professor, Department of Biotechnology, Bishop Heber College, Trichy. In this video tutorial, we will be going to see about an important mechanism called conjugation in bacteria. Right? What is conjugation in bacteria? Let me explain it very simply. So, throughout this video lecture, you will be going to learn what is conjugation, the steps in conjugation, and how conjugation plays a major role in bacterial recombination, right? So, conjugation refers to direct cell to cell genetic exchange mechanism. This is the process that allow the transfer of DNA, usually a plasmid, from a donor cell to a recipient cell through physical connection. Another important thing to be noted here is this mechanism. Conjugation is otherwise called as horizontal gene transfer in bacteria. This enables the organism to spread important traits such as antibiotic resistant genes. Right? So, with this brief introduction, let me move on to the next slide. You probably know the structure of bacteria, that it has a strong, a thick cell wall. The cell wall, another very next layer to the cell wall is cell membrane, you know that, and the blue color layer, right? The light blue color layer, what I have referred here is, what I have given here is cytoplasm, right? Cytoplasm, and in the cytoplasm, there is a tangled mass of nucleic acid called chromosomal DNA is present, right? This is a very simple architecture. We used to call bacteria as prokaryotic organism, right? Right. So, in some bacteria, in addition to the chromosomal DNA, right, that is what we saw in the previous slide, in addition to chromosomal DNA, there is another DNA called plasmid DNA, which is mentioned here. This is the plasmid DNA of bacteria. This is very small in nature, and this is probably extra chromosomal double stranded DNA, right? This is to be noted. Chromosomal DNA is somewhat packed in terms of tangled mass, whereas plasmid DNA is clearly defined as extra chromosomal double stranded DNA, right? So, in addition to chromosomal DNA, there is another copy of genetic material present in bacteria. That additional copy is said to be plasmid, right? We will move on to the next slide. And while examining the plasmid very carefully, there is a region called F factor, or it is otherwise called as fertility factor. Fertility factor, right? So, this plasmid is not present in all the bacteria, but it is present only in few organisms, and this fertility factor enables the organism to form the conjugation tube or conjugation canal called pili, right? Pili is a hollow tube synthesized by a donor bacterium. Donor bacterium means which contain this plasmid, F plasmid, are called as donor, right? In short, it is referred as D, right? Remember all these things very carefully. Plasmid, and this what's that? F plasmid enable the organism to form the conjugation tube. And as I told you, these organisms are connected together by the conjugation canal said to be pili, right? So here I have given two images, two cells. This is donor, right? On the left hand side is donor, on the right hand side is recipient. See, you could notice donor has plasmid DNA, right? Donor has a plasmid DNA, whereas the recipient has no plasmid DNA, so it is called as a recipient. Or in some textbooks, or sorry, in most of the textbooks, it is given as F plus, means the organism carrying fertility factor or F plasmid. Recipient are usually referred as F minus, right? And this F factor present in the F plasmid enable the organism to form a structure, and this is called pili, right? This structure is called pili, 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 right? Remember, the structure is called pili. This will help the organism to connect with each other, because by definition we know that conjugation is a way by which organism is transferring the genetic material from one to another, right? And this bridge, right, this bridge called pili is connecting the organism together, right? Through this pili, the DNA will be transferred to the recipient cell. So from the donor, the DNA will be transferred to the recipient through this structure called pili, right? And see, the pili is growing, right? And this pili helping the organism, I mean the donor, to recognize a receptor, right? This is donor 1, this is the one is, this is called donor, and this is called donor, and this is called recipient. This pili recognizes a receptor. The recipient cell has a protein molecule on the structure, right? Remember, and this protein will be, this receptor protein will be recognized by the pili, right? And this receptor protein is present only on the surface of F minus cell, and this pili, right, it is growing and connecting the recipient cell with the donor, right? Finally, the connection is completely established. This can be clearly observed by the formation of mating bridge between donor and recipient, right? And after this is established, after this mating bridge is established between the donor and recipient, now another main important event called transfer of genetic material, because this plasmid has to be transferred to the recipient. The complete copy has to be transferred, and this transfer is is probably through the conjugation canal. And how this actually happens, right? Let me explain it in the next slide, right? So here you could see the presence of I'll zoom the presentation. So here you could find a small thread-like structure, right? A thread-like structure. This thread-like structure is called the copy of F plasmid, which is going to be transferred to the recipient cell, and this will be formed, or this process is initiated by a gene called TRA genes, right? The gene is said to be TRA. What is TRA? It is called transfer gene. Transfer gene. This gene encodes a protein for pilus formation and DNA transfer. Another important thing to be noted is called RE site. ORIT site. RET site. So RET site refers to origin of transfer site. So this is the specific site where DNA transfer begins, right? So a small nick will be formed at the double-stranded DNA called plasmid DNA near the RET site, right? After the nick is formed, a copy of plasmid DNA gradually passes through the pilus, right? You could see this in this slide, right? So transfer of genetic material through the pilus from donor to recipient, right? And here also you could see the transfer of genetic material and it is entering into the recipient cell, right? So after it reaches the recipient cell, now you see a copy is completely reached the recipient. Here you could find the presence of single stranded, right? Here also the DNA is single stranded, right? And immediately this single stranded DNA will undergo recircularization. This is to be noted, right? Recircularization. So recircularization of F plasmid means the linear, this is called the linear form of DNA, right? Linear form of DNA. As it reaches the recipient cell, it becomes circularized and the two ends will be joined by an enzyme called DNA ligase enzyme, right? So it is now linear form, and after it is recircularized, it becomes circular DNA, and this circularization is facilitated by, the two ends of the linear plasmid. Let me explain it in a little different way, right? So this imagine this is a linear DNA. So as it is entering the host, it takes circularized step. It takes a circular shape. So to attain the circular shape, this portion, this portion has to be, these two ends has to be connected so that the linear form takes a circular structure. And this structure formation is facilitated by an enzyme called DNA ligase. DNA ligase, right? So ligase is an enzyme that connects the linear form of the two ends of linear form of DNA so that the linear form of DNA takes a circular form, right? And with the help of DNA polymerase enzyme, the single stranded DNA will get double stranded. So as I have mentioned here, double stranded plasmid, right? Now the recipient cell becomes donor. You could find it. You could find the terminology donor here, right? As I have mentioned, till last slide it is mentioned as a recipient, but once the F plasmid enters recircularized and double stranded structure is formed, it becomes donor, right? And now this recipient is capable of undergoing normal multiplication as long as the cell survive in the environment. Imagine there are millions and millions of cells are generated or derived from the parents. Remember, each offspring is carrying a copy of F plasmid now, right? Initially, the bacterial population is devoid of F plasmid. As a result of transfer, every organism derived from the parent becomes F plus, right? This is the beauty. Not only beauty, this is a very big headache to the human population because by this way, antibiotic resistance genes are quickly spread among bacterial population. One single organism carrying the resistant gene can be easily transferred to its descendants through this process. This is going to be a very big headache to the human population in the year 2050, as warned by major organizations of the globe, right? Thanks for watching. Still, if you have any queries, don't forget or don't hesitate to shoot your questions to me. I'm available to clarify your doubts. Stay tuned with me. Bye bye.